Okay, so good afternoon, everybody, and thanks for being with us today. The webinar was organized as part of the implementation strategy of the Asian Knowledge Hub on sustainable soil and land management in the region that I'm briefly going to present to you. So today we are going to open the discussion on soil health assessment tool and their use in Asia. Now, if you give me a second, I will share my screen and introduce you to the Knowledge Hub on sustainable soil management. I hope you can see my screen in presentation mode. So as I said, I'm going to briefly introduce you to the framework under which this webinar was organized. So the Asian Knowledge Hub on Sustainable Soil and Land Management. The objectives of the hubs are that of facilitating the exchange of knowledge and experiences on sustainable soil and land management in the Asian region, enhance the capacity of individuals and organizations in implementing sustainable soil and land management practices, facilitate the identification of knowledge and technology gaps that can boost research and development activities, encourage the adaptation of proven methods while inspiring innovation in sustainable soil and land management, and not last, the hub was developed and launched to support project formulation and implementation activities. Because of its objectives, the hub is seen as a driver for positive change in agricultural and environmental practices. And more concretely, it reports and describes countries' original engagements to assess, monitor, preserve, restore, or enhance soil health over time, as well as United Nations contributions on the topic. How we collected information? Well, we did it in using a participatory approach that involved the public sector, non-profit sector, academic and research institutions, and intergovernmental organizations. However, the hub is meant to support the work of a large range of stakeholders. So being an open access live tool, if you are interested in contributing to it, I would very much like to hear on your experiences and the tools that you developed in your country. So you can send me an email at the email address you see on screen, and I will be able to discuss the inclusion of your work in the regional hub. The online platform that will host the hub is still under development, but as you can see, the material in the hub is made available online already through actually these webinar series that we are running, and through other means that I will tell you about at the end of the webinar. For your information, the content of the hub is organized into 5 categories depending on their core scope. So scope 1 is on soil assessment, monitoring, and mapping. So you will find all tools related to these, so including soil sampling, soil analysis, data analysis, and interpretation and digital soil mapping under scope 1. Under scope 2, you will find tools related to policy. Under scope 3, you will find tools related to investment. Scope 4 is for field activities, and this includes capacity building activities and transfer of knowledge, data and information, and also technology. Under scope 5, you have everything related to communication. So looking at the specific objectives of this webinar, that are on promoting the use of soil health assessment tools for sustainable management and monitoring purposes in the region, assist countries in reporting on international targets, and not last, to promote the use not only of FAO tools, but also of countries' developed tools or in the independent organizations' developed tools in projects and national programs initiatives. So this webinar falls under scope 1 on soil assessment, monitoring, and mapping of the Knowledge Hub. Okay, this was all from my side. As you can see, a brief presentation on the context under which this webinar is organized. And now, for really introducing the topic under discussion today, I would like to give the floor to the first speaker, Or actually, first, let me ask you some question, because this webinar is meant to be interactive. So I would like to encourage you once again to interact with the panelists using the question and answer box, but also with other participants using the chat box. And indeed, like before giving the floor to the first speaker, that is Ms. Norquick, I would like to ask you a few questions that will help breaking the ice. So in front of you, some windows should open, and you should be able to answer a few questions. The first question that I'm going to ask you today is the following here. Can you see the window in front of you with the related question? The question is the following: how important is to assess soil health in your opinion? Little important, fairly important, or very important? I will keep these screens active for some 20 seconds each to give you the opportunity to answer. So please, Harry, usually I close them once we reach the majority. So how important is to assess soil health in your opinion? And these bring me to ask you, why are you here today? To learn how to better assess your soil health because you consider it very important, or to learn more about soil health, maybe to change your mind and give it a bigger importance? Okay, I'm closing the poll now. The majority of you said that it's very important to assess soil health. Very good. Let me ask you another question. The next question is the following: what would you focus on when thinking about assessing soil health? This is a multiple choice answer, so you can have more than one preferences. So would you say that when you assess or think about assessing soil health, you look into soil physical properties, including, for example, water infiltration rate? Or maybe you look into soil chemical properties, looking at nutrients and soil organic matter? Or what about soil biological properties, including plants, pests, or pollutants, or any other? So what would you focus on when thinking about assessing soil health? And please, if you select other, can you please tell us in the chat box, what do you mean by other? I will close the poll shortly, so please hurry up. Okay, I'm closing the poll. So here we go with the results. Most of you talk about think about soil chemical properties, but also biological properties, and we still have a good rate of people that think about physical properties. And also pollutants were not doing too bad. For those of you that said others, can you please tell us what you mean by other in the chat box so that we can consider it for discussion? Thank you very much. Now let me move to the third poll. Okay, thirdly, I would like to ask you, how do you usually assess soil health, no? So first we look into the importance of soil health, then thinking about soil health. Now practically tell us, how do you assess soil health? Do you use laboratory analysis, remote sensing, soil test kits? You do visual assessment, or you do it, you assess it indirectly by observing plant growth, or others? Again, if others, can you please tell us about it in the chat box? And note that this is a multiple choice, so you can have more than one preferences. I will close the poll shortly, so please hurry up. Okay, closing the poll now. Okay, the majority of you do it by using laboratory analysis. Also, soil test kits are not doing bad. And the same for visual assessment and plant growth. And then we have remote sensing, no, indirectly, and then remote sensing and others. Very good. Thank you. 2 more questions to go before the presentations. So the next question is the following. How often would you recommend to assess soil health by laboratory analysis? Regularly, once per week, or once per month, maybe once per year, or every time signs of soil degradation appears, or maybe only when plants do not grow as well as expected, or others. Again, if others, please tell us what other do you refer to. This is a multiple choice question because, for example, you might do it like once per month, and eventually, for example, when plants do not grow as well as expected. But please tell us. I will close the poll in a few seconds. Please hurry up to tell us your preferences if you didn't do it already. Okay. So the majority said that they do it if plants do not grow as well as expected. Still, you tend to do it at least once per year, and when signs of degradation occurs. And then we have a same rate of once per month and others. Please tell us about others. I'm curious. Last question. Get ready. Tomorrow we will have some other polls at the very end of the webinar to see if we changed eventually your approach and attitude to the topic. So how often would you recommend to assess soil health by visual assessment? So the previous question was on laboratory. This one is about visual assessment. The options are the same. So regularly, once per week or once per month, once per year, every time signs of soil degradation appears, if plants do not grow as well as expected, or other. How often would you recommend to assess soil health by visual assessment? I am about to close the poll. Please hurry. Okay. Let's see. So if plants do not grow as well as expected, if signs of degradation appears once per year, once per month, and this time also we have once per week and others. Very good. Thanks for telling us. Very interesting to know. Now, finally, it's a pleasure for me to give the floor to the first speaker of the day, that is Miss Aranya Norqui, soil surveyor at the Land Development Department, Ministry of Agriculture and Cooperatives, Kingdom of Thailand, that will introduce us to the soil health and soil assessment tools. Ms. Norqui, the floor is yours. Thanks for being with us today.
Thank you, Lucrecia. I would like to share my slides right now. Oh, I'm not sure. Do you see my screen? Can you see my slides? Okay, here. I know. I don't know what happened right now. Um, can you share my slide for me, please? My PDF file.
Yes, I will do it. You want the
Thank you,
PDF?
Kesha. Yes, PDF. Sure.
No problem. So, okay, just a second. Okay, can you see them?
Yes.
Okay.
Can you put it on this full screen or something?
Okay.
Yep. Thank you very much, Lucrecia. And good afternoon, everyone. Again, my name is Saranya Norgel. I'm also a staff of the Center of Excellence for Soil Research in Asia, or CESFA. Today I'm going to share insight on the topic, Introduction to Soil Health and Soil Health Assessment Tools. Next, please. Here is a quick overview of what we will be discussing today. I will cover the definition and significance of soil health, and also looking at the method and key indicator that provide insight of soil health. And the last one, the example of how this assessment is practically applied. Next, please. So what is soil health? The Natural Resources Conservation Service, or NRCS, defines soil health as the continued capacity of soil to function as a vital living ecosystem that sustains plant and animal and human. For most of us, soil health is a characteristic that allows to get function and specific service ecosystem. So we consider that physiochemical parameter together with biological properties are in the core center of how we measure soil health globally. Next, please. So what function do you expect your soil to perform? Our soil is dynamic and diverse natural system that provide many essential ecosystem service, such as soil acts as a cornerstone for food and habitat provision. Also be a key player in nutrient cycling and contribute significantly to carbon sequestration. Soil purify water to maintain the quality of water resources, as well as maintenance of biological productivity and environmental quality. Next, please. To measure soil health, why do we have to act now? As soil require attention due to population growth, deforestation, and climate change, creating significant pressure on soil resources. This results in soil degradation, causing biodiversity loss, nutrient imbalance, and acidification. Threatening aspects such as food security, water supply, and reduced ecosystem services. Therefore, it is crucial to assess soil health and prompt action is required. Next, please. So how do we measure soil health? It cannot be measured or determined by measuring only crop yield, water quality, or any other single outcome. Instead, soil health is assessed by formulating a holistic soil health index, composed of key soil attributes like all physical, chemical, and biological properties that help to evaluate how well the soil performs its function in various field or ecosystem related to different economic and management practices. Next, please. For assessment tool, in this case, we are looking at soil health index. Soil health assessment indices and tools have been developed since the 90s, as assisting in decision making, sustainable agricultural and management. For a robust soil health index depends on its ability to detect changes in soil management practices. Also, the use of standard soil sampling and analysis techniques, including to the statistical, mathematical model, and having a hypothesis to select appropriate soil attributes. Next, please. In general, there are 3 steps to developing a soil health index. For the first step, involved with the selection of appropriate indicator for the minimum data set. In this step, statistical approach, such as principal component analysis, ANOVA, and correlation, are commonly used to identify the most relevant and sensitive indicators related to soil function. Another approach is to consider expert opinion as an alternative method. For the second step, involve developing indicator scoring functions to convert each indicator value into a numerical or categorical type, expressing its relative status. For statistical approach, determine this relative value by utilizing linear or nonlinear functions, which rely on the assumption of a normal distribution. For the final step is to integrate the relative contribution of each indicator and then calculate an overall soil health index or score. The most common statistical tool for this purpose is the additive. Other approach aim to integrate a weighted sum of indicators or attribute specific indicator to aggregate soil function. Next, please. The useful soil health indicator should be easily measured and cost effective, also sensitive enough to detect change in soil function, be a combination of physical, chemical, and biological properties and processes. The indicator used by different research or different region may not be the same due to the purpose-oriented and site-specifics. Additionally, they should be stable in short-term weather pattern to enable the accurate measurement. Next, please. Soil health indicator for selection the appropriate soil health indicator and the construction of soil health index related to soil function or threat. Many common indicators are for physical, we can see like bulk density, for site distribution, aggregate stability, saturated hydraulic conductivity, available water capacity, and infiltration. For biological properties, for chemical property, we have pH, electrical conductivity, cation exchange capacity, organic matter, and available nutrients. For biological property, we have PMN, or potentially mineralizable nitrogen, active carbon, microbial biomass and respiration, fatty acid, enzyme activity, microbial biodiversity, nematode community, and earthworm population. Next, please. For the implementation of the assessment tool, for the case study in Thailand, soil health assessment framework have been developed using a simple method, mostly by visual and in the field, emphasizing the enhancement of knowledge and understanding of soil resources. This approach aims to enable farmers to manage and improve soil health through proper soil management. This method tends to be local in geographical scope and sometimes specific to production system. Some studies have shown that a small set of carefully selected indicators can indeed provide adequate information for land management decision. Next, please. For the methodology, after selecting the area, farmer can complete the soil health assessment form, providing physical, chemical, and biological information on soil texture, structure, gravel content, plant characteristic, organic matter, pH, EC, micronutrient, and the last one is a microorganism, according to the provide guideline. The scores obtained will contribute to the final assessment result. Next, please. Another global level implementation that I know, as a report by the 11th Plenary Assembly, indicate that GSP has been developed the final draft of Global Partnership Performance Indicator System and Soil Stat, which is a system for monitoring, forecasting, and reporting periodically on the status of global soil resources. And also will create global soil health dashboard in the very near future. This also include indicator fact sheet, operational aspect, data policy, and etc. The plan is use 15 of 21 indicators for the global soil health index, with a focus on agricultural land. Next, please. And I think that is it for my part. Thank you for your kind attention, and I'm happy to answer any questions that you may have.
Thank you very much, Saranya, for the very good presentation, and also thank you for recalling that the GSP is working on these important tools. Let me see if I can go back. Yes, this one that you mentioned, the Soil Health Index and the Global Soil Health Dashboard that we are all looking forward to have released as soon as possible. So thank you very much for recalling it. And I'm kindly asking you to please stay tuned and have a look at the question and answer control on your screen. And in this regard, I would like to remind participants to please ask any question you might have, or make any sound intervention in the question and answer box, so that we can keep track on it and our panelists can promptly reply to your questions. Thanks again, Saranya. Now I would like to pass the floor to Ms. Carolina Oliveira Sanchez, Senior Consultant on Sustainable Soil Management at the Global Soil Partnership of FAO, that will tell us about the protocol for the assessment of sustainable soil management released in recent years by FAO. Carolina, the floor is yours.
Thank you, Lucrecia, and thank you for the invitation. Hello, everyone, and I am very happy to be in this interesting space of the Asian Knowledge Hub. Let me please share my screen. in that presentation, I will talk, as Lucrecia said, about the protocol for the assessment of sustainable soil management. Okay. Okay, so the protocol for the assessment of sustainable soil management was developed by the Global Soil Partnership in FAO. It is a tool to support the implementation of the voluntary guidelines for sustainable soil management that you perhaps know. I will discuss that more in detail. The first version of this protocol was endorsed in September 2020. This first version was describing was including the description of some sustainable soil management indicators. We differentiate the sustainable soil management indicators from the soil health indicators, and this is what I will explain to you today. The process of developing this protocol was a participatory process. We had a committee composed by different intervenants from all the regions of the world, and this is how the indicators were selected. After the selection of these indicators, we have been working on the instructions, let's say the instructions on how to implement the protocol in the field. This document is in addition in this moment and will be published very soon. So what is published in this moment is the document that you see in the screen is describing only the indicators but is not detailed about how to implement it in the field. So this was the first version that you can find in the website of the Global Soil Partnership. What is the Sustainable Soil Management Protocol? Basically, it was developed to monitoring sustainable soil management compliance, the compliance to the voluntary guidelines for sustainable soil management. This means that what we can know through this protocol is if the soil has been managed sustainably or not. So this is different from a soil health index that is determining if a soil is healthy or not. Here, with the Sustainable Soil Management Protocol, we are evaluating if the soil has been managed sustainably or not. How we are doing that? We are doing that through the definition of soil health first. So the soil health is the ability of the soil to sustain the productivity, diversity, and environmental services of terrestrial ecosystems. This was the definition by ITPS in 2020. And here we find the definition from the voluntary guidelines for sustainable soil management, which is soil management is sustainable if the services provided by soil are maintained or enhanced without significantly impairing either the soil functions that enable those services or biodiversity. So this is the concept that we are working on from this sustainable soil management protocol, is to evaluate if the practices that we are implementing help or enhance the services provided by soils. So as we saw in the previous presentation, soil functions or soil ecosystem services are defining soil health and are defining also the functions that we expect from the implementation of sustainable soil management practices. In this case, we have extracted 4 main functions that we want to evaluate through the Sustainable Soil Management Protocol, providing these soil ecosystem services. So it is the provision of food, achieving food security. Second one is the mitigation and adaptation to climate change. It means the soil organic carbon that you can find in the soil. The third one is the provision of clean water. So this is through the soil physical properties and more specifically through bulk density. And the fourth is preserving soil biodiversity. So we concentrate on those 4 soil ecosystem services, and we arrive to the definition of 4 main indicators. These indicators, as we saw also in the previous presentation, are sensitive and are measurable and are reflecting changes in soil. It doesn't mean that this defines soil health. No, this is the difference between a sustainable soil management indicator and a soil health indicator. The sustainable soil management indicator is showing if a practice implemented in the field has had some impact on soils on these 4 indicators that we selected. So those indicators are soil productivity. It is measured on ton by hectare by year. It can be the yield of a crop, but it can also be the biomass produced during the year. The second indicator is soil organic carbon, that, as you know, is a good indicator for, well, climate change, but also for all the properties in soils. It is also used, this soil organic carbon is used, for example, in the UNCCD convention to evaluate soil degradation. So this, of course, we maintain this soil organic carbon percentage, well, in this case. For the physical properties, we have selected soil bulk density, as it was also shown in the previous presentation. Soil bulk density reflects the soil porosity, reflects more or less the status of soil compaction, and can be a good indicator if sustainable soil management is being implemented or not. But in this case, and in the case of all these indicators, it takes time to see the changes, no? So this is where we are going to see on how to implement the protocol. And the fourth indicator, the biological indicator, let's say, is the soil respiration rate. Why soil respiration rate was selected as a biological indicator? Because it is measurable, easily measurable. It is measured in the field, but also in laboratory. And we have some reference values to compare. This is also important. We recommend the soil respiration rate to be associated with an indication of biodiversity. Soil respiration rate is an indicator of soil activity, of biological activity, but we recommend to associate this with a biodiversity index, like, for example, the enzymes of the soil or the macroinvertebrates, etc. To these main indicators, we associate some additional indicators. If soil degradation is caused by specific or unidentified threats, the latter can be measured using additional indicators. This is the case, for example, if you see the pictures here of soil erosion. If you have soil erosion, sustainable soil management can be measured by the difference of soil erosion after implementing sustainable soil management practices. It is also the case of soil salinity, soil biological activity, soil biodiversity. It is also the case, for example, here you can see a test of infiltration rate if the soil is compacted, or the measurement of penetration rate in the soil. So these additional indicators, as it is written here, it can be used in cases where a specific soil degradation is identified. Otherwise, we recommend only with the 4 main indicators. This should be sufficient to assess if a practice is sustainable or not. How to implement the protocol? We have 3 different steps followed by their corresponding activities, and the methods are described in the protocol. So the 3 steps are the description of the study area first, then the data collection for being measured in the laboratory, and the third step is the final assessment of the indicators, the final result. So all the methods are described in the annexes, well, in the documents of the protocol, and this allows us to contribute to soil information systems. Also, we rely a lot on laboratory network and the reliability of the laboratory measurements. And this is useful for decision making, for project evaluation, and also for reporting on land non-degradation neutrality on the convention of the UNCCD, for example. So how we implement the sustainable soil management protocol? We start describing the study area. The study area needs to be homogeneous. It needs to be defined by the description or assessment units. We have, well, of course, the description of all these details in the protocol, but the principle is that the units where we are developing the assessment are homogeneous. We describe the current soil management practices that needs to be the same for all the study area. We differentiate the different units that are included in this study area. And then we are ready for doing the sampling for measuring the laboratory, the indicators data. For the data collection, the sampling design is based, as I told you, on homogeneity and the required resolution that we need for this study. So we have here two principles. The first principle is that we need to compare a baseline with the assessment time. It means after implementing the practices, how this is going, the soil is going to react, how the impact will be on soils. Here you see, for example, some aerial pictures taken in the same place before and after implementing sustainable soil management practices. So you see one picture in 2008 and one picture in 2018, 10 years after. So as I told you, the changes on soils can take time. So we cannot measure changes in 1 year or 2 years. We need to wait for at minimum 4 years for seeing changes in those 4 indicators of soils. No, so in soil productivity, soil organic carbon, bulk density, and respiration rate. So you compare the baseline with the soils after minimum 4 years of implementing practices. The next principle is that the measurement or the area needs to be homogeneous. And how we evaluate the homogeneity of this area, we proceed by implementing the visual soil assessment tools. Visual soil assessments have been published by FAO. I think Lucrecia had included that in the reference in the documents of this webinar. So we use the visual soil assessment to make sure that of the homogeneity of the study area and of the measurement area that we have. You see here a diagram in the way this homogeneity is being measured and the visual soil assessment is being implemented. We exclude some what we call bad spots, the spots that don't correspond to the general, the average data of visual soil assessment in the field. And we take 5 points with 2 samples per each point to evaluate this homogeneity. And the visual soil assessment allow us to not only to validate this homogeneity, also these data are taken into account in the analysis of the sustainable soil management, and they allow us to collect representative samples. The samples are taken in these 5 points, are put together, so the sample is a composite sample that we send to the laboratory. So this is the step of data collection. This step is common also with the protocol of soil recarbonization. We also use this protocol into the measurement of soil recarbonization that you will see tomorrow, I think. So for the data collection, we sent the the samples to the laboratory. The 4 indicators are measured in the laboratory, following the standard operational procedures defined by Glossoland, the global soil laboratory network. These are the chemical, physical, and biological parameters, as I already told you, the 4 indicators determined by the SSM protocol. And also additional indicators in relation to soil threats. These additional indicators can be measured in the field or can be measured in laboratory, depending on the threat that we are considering. For the visual soil assessment, I give you here some details. We extracted from this publication of FAO about visual soil assessment. We have extracted some of the parameters recommended by this publication. We measure, for example, the roots. Well, we assess the roots, the macroinvertebrates, the color and mottling of the soil to evaluate the drainage and water circulation and the soil structure, for example. The results of the visual soil assessment are presented by poor, medium, or good. It is a very simple way of assessing soil. And we use, here you can see some descriptions, some cards developed through the Global Soil Doctors Program, where we describe step by step these practices of visual soil assessment recommended by FAO. We describe which is the principle, which are the materials needed, the step by step, and how to evaluate if they are poor, medium, or good. So here you can see, for example, the visual soil assessment of the roots observation, and also the soil structure. So you can find these simplified sheets in the website of the Soil Doctors Program. We use it in the protocol for SSM, and we use it, as I was telling you, during the process of taking the samples and of evaluation of the study area. The final assessment of the SSM protocol need to take a main principle into account. It is the fact that the 4 indicators need to be considered together. We cannot consider one soil indicator apart from the other. It means that if you have a higher soil organic carbon, you cannot say that the practices were sustainable. You need to consider soil organic carbon together with the other indicators. So we evaluate the trend, the comparison to the baseline. If we consider that soil is not sustainably managed, if one or more indicators show a worsening trend, we consider that the sustainable soil management has been implemented in a certain measure if all indicators sorry, maintain the same values. We have also medium compliance. It means that medium compliance to sustainable soil management, or high compliance. So the high compliance of sustainable soil management, it means that management is being sustainable for soils, is if all indicators are improving. It means that you have more soil organic carbon, you have also a bulk density, a soil less compacted. It means a higher bulk density, lower the bulk density, sorry. And compared also to soil productivity, higher soil productivity and higher soil respiration rate. If these 4 indicators are all improving in the same time, we can consider really that soil has been managed in a sustainable manner. So this is the final assessment. Of course, you have more details in the publication. Here you have a summarized idea on how doing the assessment. And And this is the way that we recommend to implement the Sustainable Soil Management Protocol. How and why we implement the Sustainable Management Protocol? We, as you see here, is a mechanism working together between different topics or different teams. You need to have a good network on laboratories. You need to have harmonized methodologies, but you also need to have a good soil monitoring system, which will allow you to follow the changes on the soil. So why or in which cases we use the Sustainable Soil Management Protocol? We use it, as I was telling you, in the RecSoil protocol, in the RecSoil framework for measuring if recarbonization of soil has been effective or not. We use those indicators also on project evaluation, impact indicators, for example, for JEF projects or for projects in international cooperation in general. This can allow you to measure if the practices that you have recommended are sustainable or not. And we use also the Sustainable Soil Management Protocol in LDN reporting for the UNCCD Convention of the United Nations. So well, this is what I wanted to show you today. If you have any question, I will be here available. You have also here my email if you want to send me questions by email. Thank you very much.
Thank you very much, Carolina. And indeed, there are so many questions for you in the question and answer box. And also, there is one that is not in the question and answer box, but is in the chat. So let me ask this one live, since we have still 5 minutes to take questions live. Soil productivity, is it influenced by other indicators, or not? Because you put it as an indicator, right? In the protocol.
Yes, soil productivity is included in the protocol as an indicator for sustainable soil management. It needs to be considered together, for example, with physical properties, because you can have the increase of soil productivity can be linked to some biological or chemical properties, but this doesn't mean that your soil, necessarily that your soil is being managed sustainably. So if you need to associate soil productivity with the other properties of soils, like, for example, bulk density, that can allow you to know if soil is more compacted or less compacted than in the beginning, and also with the soil organic carbon.
Thank you very much. So as I said, there are a few answers for you, a few questions for you in the question and answer box. Is there anyone that would like to take the floor and ask any question live to Carolina, or share their experience, maybe using the protocol if you already knew about it, or any other tool related to it? If you like to do so, we have 5 minutes for this interaction. I saw some hands up and down coming. I don't know if there is still someone willing to take the floor. Yes, so we have someone. Jegan Sekar, I'm allowing you to talk right now. You should be able to unmute yourself.
Hi, I think the presentation was nice. So just an understanding, you have mentioned 4 indicators, which is crucial, which covers soil activity, either biological activity or crop activity. So what's the range? If any range defines the quality, you mean the indicator, means respiration, this much range, particularly related to the type of crop of cultivation. That's my question. Because health is fine, but there will be a level and with respect to the crop of cultivation. So can you just enlighten on that aspect?
Yes,
Thank
thank
you.
you. Thank you very much for your question. Yes, this is indeed a very good question, and a question that we have been having when we were developing the protocol. The fact is that we compare the soils through we start from a baseline. We maintain the same land use of the soil, the same crop. If you have been cultivating maize, for example, you start doing the baseline, you continue cultivating maize during all these years. And after 4 years, you measure again the indicators, the 4 indicators. But when you have, for example, changed your fertilization plan, so you have been cultivating maize, you have changed your implementation plan, you have perhaps reduced some chemical fertilizers, you have included some organic matter, and you want to see at the end of the 4 years how your soil has reacted to this change of practice. So you compare a baseline to an end line. So this is how you can have references, reference values. Otherwise, if, for example, in respiration, as you were saying, in respiration, we don't have many reference values, even if it's the most measured indicator, biological indicator. You need to have a reference value for the climate, for the specific crop, for the specific climate, and for the specific soil where you are acting. This is why we recommend to compare the baseline to the end line. And this allows us to to work without reference data, because we cannot have reference data for each type of soil, each type of climate where you are implementing the protocol. This is the reason why.
Yeah, thank you.
Thanks to you.
Thank you very much, Carolina. Now, before giving the floor to the next speaker of the day, I would just like, of course, to ask you, Carolina, if you can please have a look at the questions that were asked in the question and answer box. There is one question that came to my attention. It was addressed to Saranya, but she cannot answer to it because the work on the soil health index is held and done by the Global Soil Partnership, and actually not even by Carolina. So this is just for the information of Sarazia that asked the question, but of you all. I already contacted the person that is in charge of this work. He is currently busy, but by tomorrow I should have an answer to the question on the soil health index and related indicators, so the dashboard. And if not today, I will let you know the answer tomorrow. Thank you again for being so active. Carolina, I leave you to answer the question online. And I would like to remind you all that you can read the answer in what is called answered. Now, if you go to question and answer, you have open, answered, and dismissed question. So if you have a look at answered, maybe you will also find answers to the questions that you would like to pose. Said that, I would like now to give the floor to the next speaker of the day, that is Mr. Alexis Toumasiou from the French Agricultural Research Center for International Development, also known as CIRAD. And I apologize for my terrible French pronunciation of your last name. Alexis will tell us about the BioFunk tool that CIRAD and IRD developed in collaboration with Thailand. So Alexis, the floor is yours. Thanks so much.
Thank you, Lucrecia, for introducing. Thank you, everyone, for being here today. I hope you can see my slide. Is it in full screen now? No, you see the other one.
We can see them, yes.
Okay, perfect. You can see in full screen, right?
Yes.
Okay, perfect. So thank you. I will do my presentation today on BioFunk tool. So I don't know if you've heard about this set of indicator, but BioFunk tool stands for a set of indicator to quantify the impact of land management on soil health. And as Lucrecia introduced before, this set of tool has been developed by various institutions. And initially it was created in Thailand with a partnership with the Land Development Department, together with my colleague Alain Broman. So, well, we are all here together today on soil health assessment. So we know that there is a key issue and a key concern on assessing the soil health. And soil is really linked to humans, actually. Soil is providing many soil ecosystem services that has been also introduced before by my colleagues. And humans, on the other hand, tends to degrade these soil compartments, and especially intensive management practices and land use changes have key impacts on the soil function. So under this context, there is a key need to develop and to monitor the soil. And nowadays, we can see that there is more and more initiative and more programs that aim to develop and to better understand how management practices can impact the soil. So the definition of soil health is quite complicated. Actually, I went back to the literature and there are so many definitions, but the one we stick by since the beginning is the one of Doran and Zeiss. And actually, those authors really keep a highlight on the capacity of the soil to function as a vital living system, etc. But the point on the functioning is really important on the first definition of the soil. And actually, on the other hand, when we look at the current soil health assessment, we can illustrate it with this dog, this big dog. Why? Because most of the assessment program of the soil health aim at summing independent components of the soil, such as physical, chemical, biological properties. And the idea will be the more there is, the more there are chemicals, the more there is earthworm, the more there will be bacteria or other things, the better the soil will be. So actually, it's really gathering independent components and trying to add them up and to try to see, okay, the soil that would be the healthier would be the one who that would have more of these stocks or those properties. But when we come back to the definition of Doran and Zeiss, who highlight the need to measure the capacity of soil to function, actually our focus, rather than being on the soil properties and independent properties, would be linked to the soil function that you can have on my right. And those soil functions are mainly driven by biological component of the soil. And they can be divided into 4 main functions as defined by Kibble-Watch, which are the carbon transformation, the nutrient cycling, the soil the structure maintenance, and the pest regulation. And according to the definition of Doran, this is really what we need to focus on, those functions. To be able to assess those functions, it's not so easy. Why? Because those functions, as I said before, are driven by biological assemblage. And those biological assemblage in the soil are really diverse, as you may know, very small, very big, very diverse in terms of the different assemblages. And they are the ones who actually drive most of the functioning of the soil. Here, I put some example. For example, earthworm will dig some galleries in the soil, and then it will help or not the maintenance of the structure of the soil. Fungi, for example, there will be mycelium in the soil, and then we know that mycelium have a key impact on the soil aggregation, etc., etc. But the problem of this is that we know that soil, all those soil animals or biological components interact with each other. So they will compete, they will facilitate everything. And this interaction will lead to the functioning of the soil. If a system is very active in terms of biological assemblage, the functioning will be then very efficient. So when we want to know about the soil functions, one way that we could implement is to try to understand all these soil compartments, trying to understand all the diversity of the macrofauna, microfauna, everything, trying to understand all those interactions. But actually, it's very complicated and very costly. So the idea of BioFunkTool was to forget about all this diversity, interaction, and everything, to consider the soil as a black box, but to focus on the functions directly. So we will directly measure the soil functions, and those soil functions are actually the result of the interaction of all the organisms in the soil. So rather than measuring the stocks or the properties of the soil, we will measure how the soil is active and how the interactions of the biological component of the soil are they intense or are they reduced. So from this approach of the soil, based on stocks and based on independent properties, we wanted to focus on the integrative approach as defined by Kibble-White, which is rather the assessment of the soil functions, which are the result of the interaction of all the biotic components of the soil. So instead of knowing who is there and in what is the quantity of those organisms, what is the quantity of those stocks, we will rather focus on how they interact with each other and what is the result of their interactions. And with this, we stick more to the definition of Doran, focusing on the soil function. So this is the concept of BioFunkTool, the framework of BioFunkTool, and our objective is thus to be able to target soil functions linked to biological assemblage, and we will focus on indicators linked to 3 soil functions. We didn't integrate the pest regulation yet because it's really plant dependent, so it's very difficult to provide a set of indicators that are that can be adapted to all cropping systems. And we stick on 3 different ones: carbon transformation, nutrient cycling, and soil structure maintenance. And with this, I would like to say theoretical aspects about how to measure soil health. We try to find some indicators that can actually meet our goal to be able to measure those soil functions. To do so, we implemented 2 different steps. The first one is a literature review. So we screened all the indicators or measurements in the literature that could meet our expectation to be able to measure the soil functions. And we also organized a workshop in Thailand in 2015 in partnership with the Land Development Department of Thailand. And the idea was to gather some experts of different disciplines in soil science and to try to find some key indicators that could be used for this BioFunkTool. So the 3 criteria that we had, the first one is really to be able to reflect one of these 3 soil functions. So all the indicators should be linked to the functioning of the system and the result of the interaction between all soil organisms and activity. So, for example, we remove the soil organic carbon content because it's a stock. It's not at all measurement of the activity of the system. We also removed all the measurement of the pure diversity because diversity doesn't mean activity. Actually. The second criteria we used is to try to mainly focus on in-field assessments. The idea was not that we like to go to the field, but the idea was to focus on the performance of the soil. So to try to catch how the soil performs in situ and not in the laboratory, because the problem of the laboratory is that when we make measurements on the soil, we will disturb it, we will grind it, we will sieve it, we will dry it and everything. And this is like a potential. And under our goal to measure the interaction of the soil system, we really wanted to focus on the actual performance. And then we tried to find some indicators that are what we call low-tech. So it means cost-effective, time-effective as well. And the idea of this is that we know that measuring soil health is really heterogeneous in terms of spatial measurements. So we wanted to be able to make many replicates in the field to be able to catch this heterogeneity and to be able to understand it when we assess the soil system, rather than doing one soil sampling composite that is sometimes not really efficient to understand the plot spatial heterogeneity. And this led us to 9 selected indicators. So the indicators are linked to the 3 functions that I told you before. And here is the list of the indicators. I will not go into detail of the methodology and everything, but I'm sure there are some that you know already because they are very popular in the literature. Actually, we only developed 1 indicator, which is the measurement of the soil respiration in the field, C2X. But all the other indicators were already known in the literature and were already used for a long time. So we didn't develop indicators, but we just gathered them. The idea of having different indicators is to be able to catch the soil complexity, actually, because we know that sometimes practices will affect, for example, the infiltration of the soil, but maybe it will not have any impact on the carbon, or maybe it will have a positive impact on the carbon. So we really wanted to be able to measure all those indicators together so that we can understand the trade-offs of the synergy of the different soil functions. So we mainly work at 0 to 10 centimeter depth. And BioFONtool is only adapted to compare management practices in a given pedoclimate, so meaning same soil type and same climatic conditions, or to follow up temporal transition, as were mentioned by Carolina before, to be able to measure in one plot, to be able to measure every, I don't know, year or every 2 years, and follow up the evolution of the practice. And then we proposed a method to aggregate those results, building what we call BioFONtool index score. We can call it a soil quality index or the soil health index or whatever. The idea is just to combine all those measurements with statistical analysis, as it was also highlighted before. Statistical analysis such as PCA are good to aggregate the result. And this score is actually between 0 and 1. So meaning that we can give a score to the management practices between 0 and 1. The higher it is, the closer to 1 it is, the best the management practices would be, taking into account some expert opinion on how how optimal one indicator would be for the current comparison occurrence. So BioFONtool was initially created in Thailand in 2016, and now I wanted to show you some platforms that we built. We have a network in Southeast Asia, in Vietnam with the SFRI, in Cambodia with the Royal University of Agriculture, in Indonesia and in Australia. So we are trying to build some platform there and to have some person that can implement it and to have all the material that is required to implement the measurements. And I will show you, I hope I'm not too long, I will show you some results. I took 3 different key results that are already published, so I can send you the paper if needed. The first one is we worked on rubber plantations in Thailand, and we wanted to know the impact of different association with rubber. So we had a young rubber plantation of 4 years old and older rubber plantation of 9 years old. And those rubbers were intercrop with legume crop, such as mucuna. We had young rubber plantation with cassava as well, a control with nothing. And we had older rubber plantation with mucuna on the interrow and a control with nothing. And here are the results that we can expect from this soil health index. So you have the 5 systems below. The first thing we can observe, I make it quickly. If you want more information, I can give it to you later. The first thing that we can observe on the structure maintenance in blue is that we have a trend for an increase of the structure maintenance when we are at the young stage of the rubber plantations and when especially we have some mucuna in the interrow. So the legume cover crop actually improve the soil structure during the young stage. When we compare the old stage, those 2 plots, when we implement the mucuna in the interrow, we have an increase not on the structure, but this time on the nutrient cycling. So this can be linked to the capacity of the legume to fix the atmospheric nitrogen and to give it back to the plot. And we can also see how the carbon dynamic evolved. And we can see that the 3 young stage are lower than the 2 older stage. And this can be linked to the litter of the rubber tree that fall down every year and all the carbon dynamic that is increased. The second example I wanted to show you actually are 2 examples. We implemented under conservation agriculture, both in Cambodia and in New Caledonia. So I think you all know about conservation agriculture. The 3 pillars are minimum soil disturbance, soil cover, and crop association. It was on maize cropping systems, and it was a long-term experiment, more than 7 or 8 years, if I remember well. Yes, 7 to 8 years. So these are the results in Cambodia on the red oak soil with 70% of clay. TT is a conventional tillage with no conservation agriculture management practices, and the 3CA are conservation agriculture management practices. So you can see globally an increase of all the functions of the soil with BioFunkTool under conservation agriculture compared to conventional tillage and no biomass input. And very interestingly, in New Caledonia, which had very different pedoclimatic conditions with a much lower clay percentage and also very different soil type, we observed exactly the same trend. So maybe less, less contrasted difference, but still significant differences between the conventional tillage and the conservation agriculture system. And the last example that I wanted to show is in Indonesia. This time we worked on smallholder oil palm plantations, and we wanted to know better the impact of fertilization management practices and understory vegetation. And for this, we implemented what we call a multi-criteria approach, trying to link the management practices, so potassium fertilization and wheat biomass of the farmer. And we try to understand the impact of those two management practices on the soil health using BioFunkTool functions, and also on the oil palm performances. So we also measured some yield and the quality of the fruits and the quality of the nutrient of the leaves of the oil palm. And we try to see how management practices affect those two compartments. And what we observed is that the potassium fertilization impacted directly the oil palm trees, which this is very well known in the literature. Potassium is very important for the oil palm nutrition. And it also had a direct impact on the nutrient cycling of the soil. Wheat biomass had a positive also and significant impact on the structure of the soil. And last but not least, and very interestingly, we observed a significant and positive impact of the structure maintenance on the oil palm performances. That the hypothesis we have behind is linked to the root development of the oil palm that was better when the soil structure was also enhanced. And so in the end, we showed that the wheat biomass doesn't have any negative impact on the oil palm. That is a belief of many farmers. But in our system, actually, the wheat biomass even increased the soil structure that had some positive effect on the oil palm performance. This slide is just a summary, but I think I've been already long enough on my presentation. The idea was just to tell you that actually now we use BioFunkTool to try to better understand how agroecological management practices can enhance the soil health. And we need now to know more, to go further, and not only focusing on soil, actually, but also focusing on other compartments of the agro-system, such as the performance, the yield performance, and also socioeconomic factors that are sometimes more important than actually biophysical measurements to especially make farmers adopt to adopt such agroecological practices. Thank you for your attention. Don't hesitate if you have any questions. I've been a bit quick on the indicators, so we can discuss about it if you want, or I can share some material as well. And thank you.
Thank you very much, Alexis, for your very good presentation. Since it was the last intervention of the day, I would like to take any question from the audience live. So, because I don't think you will have the time to answer them if they ask them in the question and answer box. For now, what I can share with you is that we have an intervention from the Pacific well, of course, is thanking you for the very good presentation, and they are actually sharing their experience on working on the topic in the Pacific. So what they're saying is that in the Pacific Islands, they're also developing soil health assessment simple methods using soil physical, chemical, and biological attributes. Then they rank them and use a spider web to develop a graph, and constrictions are the attributes affecting functionality of the soil. Unfortunately, the person that made the intervention ran to another meeting, so we cannot actively exchange with him. But I think this was a very interesting case from the Pacific. I don't know if you have any opinion on what they are doing and if you are informed actually on what they are doing independently.
No, not at all, but what I can highlight is I think this comment on the spider chart is very interesting, and this is also what we try to do with BioFunkTool. Is not only to focus on carbon content or only 1 or 2 measurements, but try to gather few, like I don't know, 5, 10 or something like this, and try to understand them all instead of because this is we really see that also with BioFunkTool is that sometimes we have some hypotheses that we say, okay, sandy soil, they will infiltrate very fast. So we don't need to make measurement of infiltration. Actually, we already know the result. And sometimes from the field, when you go there, you see that actually your hypothesis is completely wrong. And actually, when you measure your soil, the infiltration was not good because for some different reasons, the management practices was done by one way or another. So we really think that it's good not to have too much like expectation on how the indicators could react, but to do it as much as possible and then to analyze the results. And having different measurements and having different variables and understand how they are linked together, if they go all together or not, is really the key aspects I think to understand soil health. And spider nest or multi-chart or something like this is a very good approach as well to do it. We use the statistical analysis, but there are many other methods that are also allowing to understand the soil complexity.
Thank you very much. BioFunkTool is attracting a lot of attention. We have people asking for your email address, so you might want to share it in the chat or in the question and answer box, or maybe give me permission to share it with all registered participants tomorrow together with the other webinar material. Also, there is interest from Timor-Leste in using BioFunkTool. I don't know, Claudino, if you want to take the floor and tell us a bit more about your work in Timor-Leste, and maybe if you have any question for Alexis. Let me give you the right to talk in case you want to take the floor.
Yes, thank you very much. I'm Claudino Ninas Nobais from FAO Timor-Leste as National Advisor for the Conservation Agriculture. Well, we are implementing a conservation agriculture in Timor-Leste for more than 10 years, and it's really providing a good contribution to increasing the crop productivity, mainly for the maize, and now we are using to different crops. We use Mucuna as best option of legumes because of produce big, huge biomass that can quickly increase the soil fertility and maybe other soil, physical soil and others, biological and others. So many farmers are willing to adopt this kind of legumes that are really, as you present, I really appreciate your presentation for the tools, BioFunkTool. I'm interested on that, how, of course, this step that you explained, that how we can discover the change of the soil from testing or from applying the conservation agriculture, but I want to know more deep on the biofunctional tool for the soil management. Thank you.
Yes, thank you for this comment. Actually, it works quite well on conservation to highlight how conservation agriculture can be good for the soil health, and it worked as I showed in Cambodia and New Caledonia. For all don't hesitate to, if you have specific questions on some method or some anything, you can contact me. But also don't hesitate to look at internet. We have a lot of publications actually more than 10 on biofilm tools. So we have also a lot of references and everything. So don't hesitate as well to have a look. And I would be very happy to share with you some idea or anything. Don't hesitate.
Okay, yeah, thank you. I can contact you by your email, and we can discuss and we can share some information.
Yes, exactly. And also, as I presented, so it was we have some platforms as well in some different area. So for me now, I'm posted in Bangkok now, so we are really having a strong collaboration with the Land Development Department of Bangkok. But we also have some different platforms, as I showed, in different places, so it can also help to share, because it also a lot of protocols, like quite some methodologies that sometimes are not so easy to implement, so you need a bit of time. And sometimes what we like to do is also to share with different platforms where you could use it. So I can also give you the contact of the people who are implementing it in our partners countries.
Thank you. Yeah.
Thank you very much, Alexis, and also Claudino for taking the floor. I don't know if anyone else would like to take the floor and make an intervention. We still have 2 minutes before closing the webinar. If not, I would proceed with the closing today's session. And let me just have a look. Okay, I'm reminding you that tomorrow after the webinar, I will share all the presentations that we're providing during today and tomorrow, and also the video recording, so that you can go through the material anytime you want. Also, we share the contacts of the panelists, so you can contact them in case of interest. Alexis, just for your information, there is a question, or actually like a comment in the question and answer box for you, if you want to have a look and eventually reply either now live or tomorrow. Better if you reply now, because I'm about to close it to close the webinar. So if you want to go through it and answer,
email, I think, if possible, just maybe very quickly for this weed species, we didn't have the opportunity to measure the diversity of the weeds and the local weeds. So I cannot answer. We only had the weed biomass, so it's study that could be implemented later on all time.
thank you.
But don't hesitate to send me email otherwise.
Thank you very much. I'm sorry, that's the downside of being the last speaker, no? You have to answer a bit in rush the question. But anyway, in case, I hope you will be with us tomorrow. So in case participants have additional question for you, they can also ask them tomorrow or by email after the webinar is implemented. So we are heading to the end of today's session that was centered on introducing the topic and getting to know about the protocol for the assessment of sustainable soil management and the BioFunk tool. So I would like to thank again all of you for your active participation and all speakers for their very good presentations. Tomorrow, we will hear about the biodiversity integrated assessment and computational tool, BINTACT, and the GSOC MRV protocol, and then discuss the possibility to use these and other tools in the region. So tomorrow there will also be the time to hear from you all on the soil assessment tools and strategies that you usually use, so that please feel free to prepare a short intervention on it if you feel like sharing with the audience. We will really give you the floor. And with this, I would like to thank you all again for being with us today, and we are looking forward to see you tomorrow as active as today at the same time. So thank you very much, and I wish you all a good continuation of your day. Bye.
Thank you. Bye-bye.
Bye.